Hi, Michelle Glass here. Welcome back to our discussion of the digestive system. We're still in chapter 24, lecture 24, and our topic here is really the star of the digestive system, the small intestine. The small intestine is where digestion is finished. It's where nutrients are absorbed from the digestive tract into the bloodstream, and we'll see that hormone production in the small intestine is really critical for the coordination of the digestive tract. Essentially, what we see happening is as chyme moves from the stomach to the small intestine will very often tell the stomach to sort of stop sending me stuff so that you can move that material through the small intestine slower, allowing time for good digestion and full absorption of nutrients before you get that next batch. And so remember I said earlier, you eat a meal, you fill up your stomach, but it takes like 4 hours to digest that meal. And the reason it's taking 4 hours is because the small intestine only allows the stomach to send it small batches at a time. So our small intestine is a big long tube and it's divided into 3 sections. So the top portion is called the duodenum or duodenum. The middle section is called the jejunum, and the very last portion is called the ileum. And of course, I've not drawn this to scale, obviously. The duodenum is kind of like a mixing bowl. So we see the chyme from the stomach, and we see the pancreas will dump digestive enzymes, and we see that the liver will dump some digestive enzymes and some bile into the duodenum. And this is going to continue the process of digestion that started in the mouth. The jejunum is really where we see digestion finishing and the absorption of those nutrients. And so we see that the jejunum area of the small intestine is folded and folded and folded again. So we are, as much as possible, increasing surface area here so that you have a lot of contact between the wall of the jejunum and the chyme, the nutrient soup that's in there. And so what we'll see is you have your simple columnar epithelial cells of your mucosa have microvilli as part of the cell membrane, and we describe that as the brush border. But those cells are positioned on top of extensions called villi. And notice that sticking up into those villi, you have what's called a lacteal. This is a lymphatic capillary. You also would have blood capillaries, which I've not drawn in here. So of course, look at the artwork that's provided in your textbook for a more detailed drawing. We can zoom in because not only do we have folding from the cells, microvilli, or these extensions of the jejunum wall called villi, but we see that we have these transverse folds known as plicae circulaires. So the whole wall of the small intestine, especially in the jejunum, is folded. And then you have villi on top of that, and then you have the microvilli at the cellular level. Now, the very first time I was teaching this material and thinking about this material, I was driving home from work, and we live in beautiful East Tennessee, and so you can see the skyline of the mountains, and it was either fall or winter, and so there were no leaves on the trees. And I saw the mountains, and then I could see the trunks of the trees, and then I could see the branches of the trees. And I thought, oh, it's like the small intestine. So in that example, the mountain would be like the fold of the small intestinal wall. That's the plicae circulaire. And then those villi would be like those individual trees sticking up. And then those microvilli would be like the branches. So if you imagined all of that connected as tissue, clearly you're increasing your surface area versus just a flat land and a flat sky, right? Okay, and then our final portion of the small intestine is the ileum. And this section is going to control the flow of material into the large intestine. And here's the thing to keep in mind, this whole digestive tube is an assembly line. So the stomach is able to hold, all of that stuff that you shove in your mouth. You sit down to eat a whole meal, you have a sandwich, you have some chips, you have some fruit, you have a drink, maybe you have dessert. So you put, a whole lunchbox full of stuff into your mouth, and all of that is sitting in the stomach. And then only little bits of that is going to move into the small intestine. And as it moves into the small intestine, it's moving along whatever was there, and it's moving from the small intestine into the large intestine. And so then, you can move along your digestive system by eating food. You see this really in babies. So for those of you who've ever taken care of an infant before, you feed them, and while they're taking the bottle or the breast, they're right away filling up their diaper with poop. So you breastfeed and you change diaper. You breastfeed and you change diaper. And so you're just seeing that the bringing in of that new food, that new milk, is pushing along the material in the digestive tract. Okay. One of the jobs, remember, of the small intestine is not just digest that food and absorb those nutrients, but let's regulate that whole system. And so we're going to look for these patterns. Essentially, our duodenal hormones, so these hormones are coming from the duodenum, are going to increase secretions from the pancreas, the liver, and the gallbladder. Now, these are accessory structures. These are releasing a lot of the important hormones and enzymes that are involved in digestion. We'll have a video just on these. So we're going to mention them a little bit here, but we'll go into some more detail in the future. Very often we see duodenal hormones decreasing the stomach activity. Remember, we don't want to push things along too fast. We want time to get a quality digestion and a quality absorption of that nutrient. And then a lot of our duodenal hormones are going to signal for our body to prepare for these nutrients we're getting ready to absorb. So if you're getting ready to give your bloodstream a lot of glucose, you need to get insulin produced so that those cells can then start the process of getting ready to take in that glucose. And then the other thing that's super interesting is that your hormones are specific to the molecular composition of the chyme. So depending on what foods you're eating, what molecules you're digesting, you're going to get a different group of hormones being released. Now, that's super interesting, but typically we're not eating food that is just one macromolecule. So most food would have a combination of lipids and proteins and carbohydrates anyway. So you would really expect to see all of these released on some level. Okay, here is our first one. Now this one is enterocrine, and this one is released when chyme enters the duodenum. So this is not specific to a macromolecule, but just the presence of chyme. And this is going to signal our submucosal glands of the small intestine to increase alkaline mucus. So this production of alkaline mucus is going to help to protect the small intestine from that acidic chyme. Remember, your stomach is producing hydrochloric acid. So the pH in your stomach is like a pH of 2, which is super strong. But the stomach is able to, it's adapted for that, so it's fine. The rest of your digestive tract is not. So if you are shooting this acidic chyme into the small intestine, we need to be able to buffer that. And we will see some buffers coming from the pancreas, but also here, we're getting our submucosal glands to make an alkaline mucus to help protect against that acidic chyme. Okay, if you have incompletely digested proteins entering the duodenum, then we have G cells in the duodenum, just like we had in the stomach, and those G cells here are releasing gastrin. So if your proteins are not fully digested, then your stomach needs to do a little bit more work. So you're going to see some gastrin being produced here. This is really the only duodenal hormone that's going to increase the activity of the stomach. So you're increasing both the motility and the hydrochloric acid and pepsin production. because there you're going to do a better job of protein digestion. Okay, next we have, again, just generic chyme, signaling the release of the hormone secretin. So here, when chyme enters the duodenum, we get the release of secretin. Secretin is going to talk to the pancreas and signal an increase in the secretion of buffers. And this is going to, again, help with regulation of the pH, dealing with that acidic chyme. So it's going to make it more basic, which is the type of environmental condition that we need in the small intestine. We'll see that secretin also talks to the liver. And it says, okay, liver, kick up your production of bile. Bile is something that's being made in the liver and then stored in the gallbladder. And we'll talk more about this in the future. Bile is going to be important in what's called emulsification of fats. So basically, it takes a big droplet of fat and it makes it a bunch of smaller droplets so that you have more contact between your lipids and your digestive enzymes. So if we have chyme coming into the duodenum, we're going to have some lipids there, so we need to get that bile production increased. And then with secretin, we're ready to just commit to digesting this chyme right now. So we need to tell the stomach to just stop, decrease the motility, decrease the secretions of the stomach, give that small intestine enough time to fully digest the nutrients here and then absorb those nutrients. Okay, our next one is called gastric inhibitory peptide, or GIP for short. This one is released by the duodenum when fats and carbohydrates, particularly glucose, are found in the small intestine in the duodenum. The gastric inhibitory peptide, as its name implies, is going to inhibit the activity of the stomach. So it's another hormone that's telling the stomach, whoa, slow down, I have enough to process right here, don't give me any more. GIP is also going to talk to the pancreatic islet cells. Remember, these are the endocrine cells of the pancreas. These cells that we're going to talk to are specifically those beta cells. The beta cells, remember, are responsible for releasing insulin. Insulin is going to be important in the absorption of glucose. Insulin is going to be important in telling your cells to take that glucose from the bloodstream and use it to make ATP. We will see it talking specifically to skeletal muscle cells and say, okay, go ahead and increase your uptake of glucose. And then because we have all this glucose available, then we're going to store any fatty acids that are being absorbed as triglyceride molecules. So we're going to tell those adipocytes of the adipose tissue to go ahead and increase lipid production. So here, insulin is not talking to the GI tract, but it's talking to the cells of the body. It's preparing them for what's getting ready to be available in the bloodstream. In the past, we have had a departmental final question on what type of cells release insulin, and then what type of cells release its opposite, which is glucagon. So make sure that you know that insulin is being produced by beta cells. And then here's a nice little memory device if you're having trouble remembering the job of insulin. Insulin is going to get glucose, and look at that, I forgot my E, glucose, into the cells. And so this is going to be released when blood glucose levels are high. I think we talked a little bit about glucose regulation in the topic of diabetes mellitus back in the endocrine chapter, but we'll do another conversation of that later in the digestive chapter. Okay, our next hormone is cholecystokinin, or CCK for short. This one is released when chyme that is particularly high in lipids and proteins enters the duodenum. CCK is going to talk to the pancreas and increase the secretion of digestive enzymes. So the pancreas is going to be making a lot of enzymes that are going to help with the breakdown of lipids and proteins and also carbohydrates. CCK also talks to the gallbladder. Remember, our gallbladder is going to store bile in the form of bile salts. So CCK will trigger the release of bile salts, which is going to aid in the lipid digestion. So if you have chyme high in lipids, then it makes sense that you want this bile available to help with the digestion of those lipids. And then here's another hormone that's going to inhibit the activity of the stomach. Again, basically, like, we've got plenty to do here, don't send us anymore. And we see, too, that this hormone, CCK, is talking to our central nervous system and helping to reduce hunger. Okay, our next hormone is called vasoactive intestinal peptide. Vasoactive intestinal peptide is abbreviated VIP. So here's a new understanding of that abbreviation. Vasoactive intestinal peptide is going to talk to the small intestine, and it's going to trigger the increase of secretion by that small intestine, so it's going to stimulate it. It's also going to talk to the intestinal capillaries, triggering dilation. So you're increasing blood flow, which makes perfect sense because, number one, we're using hormones for communication. Hormones travel in the bloodstream. So all these hormones that are being made need to get into the bloodstream and then real quickly get out to go to the stomach or the local pancreas or the local liver to signal these changes. Number two, we are getting ready to absorb a bunch of nutrients, and all of that absorption is happening across the jejunum and into the bloodstream. So we need those capillaries to have increased blood flow so it can really pick up and take those nutrients where it needs to go. And then again, see, back to this pattern, VIP is going to inhibit the activity of the stomach, slow it down. We've got plenty to do already. All right, so those were our hormones. What we want to look at next is what's happening in terms of the digestion of that material. Okay. When we are looking at our carbohydrates, our lipids, and our proteins, they all have different enzymes involved in the digestion. So here we see carbohydrates are going to trigger the release of pancreatic alpha amylase. Now, we had salivary amylase produced in the mouth by the salivary glands, but it gets denatured in the stomach because of the hydrochloric acid. Pancreatic alpha amylase is basically the same kind of hormone enzyme, except it's being produced by the pancreas, but it's still going to help break down those polysaccharides. So as we move into the jejunum, we have disaccharides and trisaccharides. Our polypeptides that we're getting from the stomach, remember, proteins were mechanically broken down in the mouth, hydrochloric acid, pepsin, and some mechanical digestion in the stomach. And so we've broken down those full proteins into polypeptides. And we will see enzymes from our pancreas important in the breakdown of proteins. We can just collectively call this group pancreatic proteases, which means enzymes breaking down proteins. Here's a specific list. I'm not going to ask you to name these specific enzymes, but you will see different enzymes used depending on what the polypeptides are. So we have trypsin and chymotrypsin and elastase and carboxypeptidases in this group here. And by the time we're getting to the jejunum, we're going to have short peptides and amino acids available. Our glycerides, our lipids, remember, started being broken down in the mouth by the lingual lipase, which continued to work while in the stomach as well. Now we get additional lipase. We get the pancreatic lipase, which is going to increase the digestion. And then we're also getting bile salts from our gallbladder. So remember, the bile is made by the liver, but it's concentrated by the gallbladder. Now, bile salts do what's called emulsification. Emulsification is taking a big drop of lipid and breaking it up into lots of smaller ones. When you do that, you're increasing your surface area. So you're increasing your contact between the lipid and the surrounding enzyme. We'll see the way that the bile does this is by forming what's called a micelle. A micelle is, similar to, almost like a cell membrane. So a bile molecule is going to have a hydrophilic and a hydrophobic part. And so it can break that big drop of oil into smaller drops because it's basically surrounding that lipid with these molecules that have the hydrophilic and the hydrophobic part. So you can see what that looks like here. By the time we're getting to the jejunum, we have broken down glycerides into the monoglycerides and the fatty acids. Okay, so now we're going into the jejunum. The jejunum is where stuff gets super folded. So I've illustrated my villi here. I've drawn in a few of those epithelial cells so that you can be reminded of the microvilli. Collectively, we're going to talk about that as the brush border. Remember, there's an additional layer of folding with those plicae circulares that I didn't include in this illustration. Notice with each of those villi, you're going to have lacteals. Lacteals are lymphatic capillaries, and you're also going to have blood capillaries. Now, the job of our jejunum is to finish digestion, and this is happening with the help of the brush border. So those cells are producing specific enzymes to specifically break down carbohydrates, lipids, and proteins. And the brush border, the epithelial cells, are going to be responsible for the absorption of nutrients. So getting it out of this nutrient soup through the mucosal cell and into the bloodstream. Let's look at our carbohydrates first. Carbohydrates are coming to the jejunum as disaccharides and monosaccharides. And what we're going to see is those monosaccharides, that's like glucose. It can go straight into the brush border mucosal cell through facilitated diffusion, and it's ready to go right into the bloodstream. Those disaccharides have to be broken down another time. So disaccharides are broken down by specific brush border enzymes. So I've listed a few here: maltase, sucrase, lactase. They're sounding just like the sugars that they break down, but they have the ASE ending to tell us that they're enzymes. So now that disaccharide is being converted to a monosaccharide. Again, you have facilitated diffusion through the mucosal cell directly into the capillary. All right, next is your proteins. So amino acids, they're ready. So if you've already broken it to an amino acid, it's going to move through either facilitated diffusion or co-transport through that mucosal cell directly into the capillary. Short peptides need to be broken down a little bit further. So we're going to have our brush border enzymes, the dipeptidases, that are going to be involved in breaking them down into the amino acids, and then of course moving them through the mucosal cell into the capillary. So pretty straightforward. Lipids are more complicated because they're lipids, they're non-polar. How are they going to travel through our polar bloodstream? So our monoglycerides and fatty acids are going to move into our mucosal cells. These cells will actually build a new triglyceride molecule and then wrap it up with proteins so that it can be transported through our polar body fluids. And that triglyceride wrapped up in proteins is called a chylomicron. And then we have a problem. Chylomicrons are huge. They cannot move from that mucosal cell directly into the capillary. So what are we going to do? How are we going to get this lipid to our body tissues? How are we going to get it into our bloodstream? Oh, I know. Let's use a lacteal. Lacteals are lymphatic capillaries. They have those endothelial cells that are acting as the one-way valves that are really making a big opening. You know, entire cells, pathogens can fit through here. So yeah, chylomicrons can fit in here too. And lymphatic vessels ultimately dump into the subclavian vein. So there we go. We're in circulation at that point. So we see that the lipids have to take the back door to get into the bloodstream. So a little bit more complicated. Okay, and that's it for now.